All right, now that we're done with how the image is formed on the retina, we want to appreciate what happens to the retina. Okay, we're now at the point where we want to turn the chemical components to give us something that we can understand, an impulse. All right, so as a review, we have the eyeball that has a lens, and the space in between the two diaphragm iris is a pupil. And then we have the cornea, we have vitreous humor, and the retina is right inside here. This is the curved surface where we usually have the image placed, and then we have the one with the most acuity called the fovea. This has more of cones, and then we also have a blind spot, okay, where there's no cones. It's a small area where the optic nerve uh, goes through. So the fovea is high concentration of cone receptors, and the periphery has predominant rods. So this is a reminder we have about 6 million cones and 12 million rods in the human eye. Um, a distribution of the rods and cones vary in different areas of the retina. For example, the fovea only has cones and no rods. And while proceeding from the fovea towards the periphery of the retina, we see that there is an increase in rods, and the cones decrease in number. And at the periphery itself, though, we see that only rods are present and cones are very sparse. We also want to remind ourselves of the structures of these rods and cones. So the rods are long and slender, and they have discs in rod cells that are close structures with rhodopsin. So if you see these, they're known as discs, okay, and within them we have rhodopsin, and they're on the outer segment. In the inner segment, it contains um, organelles with mitochondria, right? And then we have the synaptic end, and before that, we have the nuclei. Um, the cell body arises from the inner segment, passing to the outer nuclear layer through external limiting membrane. Okay, so these is now going through the layers that we already had. But what I would want you to get out of this is that in the outer plexiform layer, we have these synapsing with bipolars and horizontal cells as well, and they contain or they release glutamate. What about the cones? The cones are very similar structures, but the outer segment is small and conical. So this is the green part here, and we see that it's also got discs and these infoldings of cell membrane from formed saccus, call them saccus, and photopigment synthesized in the inner membrane, in the inner segment. Um, so the inner segment organelles have much more mitochondria, and the cell body as well has nucleus, and then we have our synaptic endings, which also release glutamate. Um, so the rods are very sensitive to light and have a low threshold. They're responsible for dim light vision or night vision, and this is known as scotopic vision. They do not take part in resolving the details of the boundaries of objects, that's acuity, or the color. Um, the vision here is just about black or white, or combination of blacks and whites. That's gray. The cones on the other side, they have a high threshold for light stimulus and very sensitive only to bright light. And they are used only for daylight vision, and this is phototopic. I beg your pardon, photopic. So for acuity of vision and color vision. So scotopic, photopic. All right, so we have three types. We have the blue cones, and we have the green cones and the red cones. Each of them sensing different parts of the spectrum, and the combination of them sensing the entire spectrum from 350 all the way to 750, inclusive that of the rods, which are the black one. All right, so let's get into some chemistry. So what we have here is the ionic basis of the potentials that we find in these receptors. Uh, we have sodium channels in the outer segment of rods and cones, okay? And these open in the dark, okay? So imagine it's dark, there's no light going in, you have sodium channels that are open, okay? And the current of sodium flows from the inner, okay? And it goes to the outer, all right? So you have sodium from the inner, then it goes to the outer, all right? Current also flows to the synaptic end. So we have sodium channels. All right, first of all, sodium channels in the outer segment of the rods and the cones that are open. So because they're open, you have current that is flowing from the inner and sodium goes out. All right, and we also have sodium also going to the synaptic end. The sodium potassium pump, which is in the inner segment, is the one that maintains this flow, so to speak. So it will release the sodium, and the sodium will go to the outer or to the synaptic end. All right, that's what this is saying. This is in the dark. We haven't yet talked about the light. So in the dark, this is what is happening. So what happens when there is light? So light will come through, and remember it's going all the way to the epithelium and striking the epithelium, which bounces back. Do you remember that black? Yeah, it absorbs some of the light that is coming through so that the photoreceptors are not overwhelmed. Okay, so some of the light will be absorbed, but the others will be reflected to the um, photoreceptors. And the first thing that will happen is that they will strike the outer segment. So what happens now? There's lights has stricken this outer segment. First of all, reactions initiated first start by closing some of the sodium channels that we talked about earlier. So sodium channels, some of them are closed down. We'll talk about how that is done very soon. So what does this result in? This results in the inside of the cell becoming less positive, right? Because initially we were having this positivity, we have been going in. It comes out, but it's going in. But now if it's just coming out and there's no going in, it makes the inside of the cell less positive or more negative or hyperpolarized, okay? So a hyperpolarized state reduces the release of whatever is happening at the synaptic junction. Now, the fact that there was uh, an amount of positivity occurring was leading into a release of neurotransmitters at the synaptic junction. But because of this hyperpolarization, that is stopped. So the reduction or the inhibition okay, of release of neurotransmitters will lead to inhibition of bipolar cells. Okay, so the bipolar cells now will not have uh, anything coming in from the photoreceptors. And ultimately, this will lead to an action potential in the ganglion, which we'll talk about very soon. Okay, so now that we have a basic idea of where we're going, let's just pause so that you go back and listen to what I said. All right, so let's talk about the closure of the sodium channels. What happens? Why close? We are going to talk about rhodopsin. Okay, now rhodopsin is a photopigment, and I told you where it is found. Now it has a protein that is called opsin. Okay, it has a pigment that is called opsin. Now, what we have here, okay, now the it's a retinol derived from the carotenoid substances like B carotene. Hence, that's why you're told to eat lots of carrots. Okay, so retinal is present in form of 11 cis retinol, known as retinine 1. Okay, so we have this retinal that is present and it is coupled to a G protein. Let me just give you a better picture of this. Just give me a minute. All right, so here's another view of rhodopsin. So, as I said, it's a photopigment, so this is showing one of the discs, and it's situated right in the disc area. And so it has opsin, right? So this is a rhodopsin molecule. It has the opsin component, and within the opsin component, the opsin also called, it's called different names in rods and cons. Right in the middle, we have a retinal. Now, the retinal is derived from carotenoid substances, as I said earlier, and this is in the form of 11 cis, okay, 11 cis, so it's known as retinine 1, okay, and retinine 1 is coupled, or this entire structure is coupled to G proteins. Another view of this that I can give you is the following. Okay, so uh, remember this is 11 cis retinol. Okay. Um, this one is showing it in light. We'll come and talk about the all trans, but this is the retinal right there. This is the rhodopsin molecule. Okay. And then maybe one more structure. Okay. That's more holistic. So we have the outer segment, it's a broad outer segments, we have the discs there, lamellae, and then we have those free floating discs right in there, and that is okay within the discs. That is where we're going to find uh, our rhodopsin. So this is showing two structures. So as I said, we have the disk, and within the layer of the disk, that's where we're going to find our rhodopsin, and it is attached to a G protein. We're going to talk about it very soon. So going back to the physiology, we see that light has the ability to change the shape of the retinal. Okay, it converts it to an O trans isomer. Now, if I can take you back just a few slides up, uh, we saw one. Okay, you've seen this O trans retinal. Okay, all right. So it has this ability to convert to an all-trans isomer, and this, okay, alters the configuration of the opsin. So how does it alter? The opsin changes. Um, the opsin once. How does it alter this? It alters this because of the change from yes to good. So the opsin change activates associated G protein. Remember that we said that these are um, associated with G proteins. So the G protein in this case is transducin. So let's see what A is showing. A is showing the opsin and an 11 cis retinal. So this is before the change. And then once light strikes, it changes the 11 cis to an O trans isomer. And once there's an O trans transition, then we have the opsin changing its configuration. And the change of configuration is the one now that activates the associated G protein transducin. This is shown in B, okay? Yeah, this is shown in B as transducin over here. Okay, so B is showing an outer segment membrane and a disc. And on the disc, that is where we find our rhodopsin. So the light stimulation of rhodopsin leads to the activation of the G protein transducin. Okay, and what does that do? The activated G protein activates CGMP. So this is a cascade that we're following. It activates CGMP phosphodiesterase, which is PDE. Once this is done, then we have PDE hydrolyzing CGMP, reducing its concentration. So we have CGMP. Once PDE is activated, it hydrolyzes this CGMP, and then we have it reduced in amounts. Okay, so what's the fuss? What's the fuss? All right, it's because CGMP keeps these sodium channels open, and once it is hydrolyzed and turned into GMP, it means that there's no more CGMP to keep these channels open. And now, because these channels are no longer open, the inside of the cell becomes hyperpolarized. Okay, so let's tell the story. Um, Before we tell the story, this one is highlighting the fact that it's the alpha subunit that activates the CGMP. Now, if we go back, okay, CGMP phosphodiesterase. Okay, have you seen this alpha? All right. So the alpha is the one that activates the PDE that then goes on to activate, to hydrolyze the CGMP. Okay. So the alpha subunit activates the PDE, and this converts CGMP to GMP. Okay. Now CGMP, as I said, it normally uh, acts directly on the sodium channels to maintain them in an open stature. And the decline of cytoplasmic CGMP will lead to these channels closing. So this will produce the hyperpolarization that we alluded to. So let's look at the whole picture. What are we talking about here? In the duck, Rhodopsin is not active. So CGMP is high in amounts, and we have our sodium channels open. So sodium coming in from the uh, internal component, okay, from the sodium potassium pumps, comes in through these channels, all right? And therefore we have this positivity that is kept up within the cell. But, and because there's this positivity kept up within the cell, we find that there's this tonic. Now, tonic is something that happens at rest, all right? Something that you can see happening at rest without any stimulation. So there's a tonic release of the neurotransmitter onto the bipolar cells. This is in the darkness. Okay, so in the darkness, we have CGMP, which has the sodium channels open, and even when the potassium pump pumps out the sodium, we have sodium coming in through the outer channel and releasing, causing the release of the neurotransmitter. And so there's a neurotransmitter that is being released onto the bipolar cells during the dark. Okay. Um, the potential is about negative 40 millivolts. Then there's the light. And the light acts on rhodopsin. They call it bleaching. Okay, it bleaches. Okay, it bleaches the pigment, and opsin. The opsin changes. The opsin activates the G. Okay, let's go back. All right, the opsin change activates the associated G protein, and then, okay, and then upon activation, the alpha component of the G protein works on the PDE. And once it works on the PDE, you have CGMP that is then hydrolyzed. Okay, let's go back. Okay, so once it hydrolyzes, then you don't have any CGMP to keep the sodium channels open. And because they're not open, now you have a situation where there's hyperpolarization. So because there's hyperpolarization, you find the membrane hyperpolarizes to about negative 70 millivolts. And what will happen is the neurotransmitter release will decrease in proportion to the amount of light. So if there is a lot of light, you will find an increase in hyperpolarization. And if there's little light, you find that there's not much hyperpolarization that is taking place. Um, we also have C, which is called a recovery phase. This recovery phase is where the retinol recombines with the opsin, okay, so that the rhodopsin goes back into a state of being um, re-stimulated. I beg your pardon for the background noise. Lots of cars going on. Okay, so what are we talking about? The rhodopsin will require, apologies, I had to cut that. But anyway, that particular slide was showing you how vitamin A, together with um, other components, help to rejuvenate our rhodopsin. But this is the resynthesis of rhodopsin. So you find that the all-trans retinal has been converted into 11-cis retinal. This is the resynthesis itself, where an enzyme known as retinal isomerase is involved. So 11-cis retinal immediately then combines with scotopsin to form rhodopsin. So let's look at how this is doing. So first of all, you have light, okay, and through several stages, we have an all trans retinal, okay, all trans retinal here. that is there due to light. And all transretinal can also change into all transretinol. And with isomerase, we have it being turned into 11 cis retinol. And this together with scotopsin then gives us back our rhodopsin. This is all done in minutes. And then once they're bleached, then they can be reused. So again, the all trans retinol is converted into 11 cis retinol by the enzyme isomerase, and then it is converted into 11 cis retinal, which combines with scotopsin to form rhodopsin. On transretinol is also reconverted into all transretinal. So it would be good for you to study all these conversions and just know how resynthesis occurs. So again, I think every time we go through a stage, you'll be going back to the steps. So light is absorbed by the photopigment, okay? Retinal and opsin dissociate. Transducin is activated. Then we have phosphodiesterase being activated. CGMP levels decrease. Sodium channels close. With less sodium entering the cell hyperpolarizes. Okay? And because there's this hyperpolarization, remember for exocytosis to occur, you have to have some calcium channels being open. And so now they are closed because there's not state of depolarization. And then we have now the transmitter release being decreased. And now the graded potential in the bipolar cells gets smaller. Okay, so this is where we are.